Hi, Bio 111 folks. I thought I would show you how to interpret your plates. Now remember, when you take a picture of your plates, you might not know what you're looking for. So I thought I would take an example from the Monday crew and show you a little bit about how things are supposed to be done. And I'll kind of go over it with you as you watch this video. So I hope it helps you. Remember, the idea was you had 4 isolates on your master plate. In this particular case, they're called A, B, C, and D. And then you had 7 R2A plates, and on each of them, we put a different bacterium. So here's what I want to tell you. We're going to start with Micrococcus to see the effect that A, B, C, and D have on Micrococcus, looking for zones of clearing. No zone of clearing is a minus. Sort of one is a plus minus. A zone of clearing is a plus, and a very large one is plus plus. I hope that's clear. You're going to be making a chart, just like I showed you in class, and I hope that it'll help you. So let's go ahead and start with Micrococcus. Sometimes it's hard to get a good picture, so I'm showing you this way so I can see exactly what you're noticing. Notice that while you might have something for A, B, C, and D have huge zones of inhibition, definitely plus plus. Remember that Micrococcus is kind of a wimpy strain. Also, remember we were diluting the bacteria and then swabbing them onto the plate. So you can see that Micrococcus doesn't like that. The next one I want to talk about is E. coli. So we're seeing what A, B, C, and D do to E. coli, and look at that. It's very clear that, well, I would say A and C definitely, and maybe D. You might get better results on Friday. Let's move on to Bacillus Subtles. And when we look at Bacillus Subtles, I would argue that C and D give zones of inhibition. Do you see them? And then we move on to Pseudomonas. Now, a lot of people didn't have anything with effects on Pseudomonas, but look at isolate C. And maybe D, but definitely C is having an effect on Pseudomonas. How about Enterobacter? Now, I want to warn you that Enterobacter formed kind of slimy stuff on the plate, so it might be a little bit hard to see. But you would agree with me that D is having a huge effect, and so is C. So I would call D a plus plus, C is a plus, and A may actually be a plus minus or a plus, but B not at all. After that is Acenetobacter, and when you look at that, maybe for C, but not so much. And then finally, Staphylococcus Epidermidis. Now I have to tell you that for the Tuesday lab, we had some weird effects on that, and I'll talk about that in a moment. But here's how things look. I see a really clear zone of inhibition for A and for C, and maybe for D. So what this means, friends, is that this particular group absolutely has 2 strains that we would want to look at. What's good about that is that we can actually have that group do, for example, A, and then what they could do is donate C to someone who didn't like their results so much. Now I want to say a little bit, I noticed this with a couple of people in the Tuesday labs. I think some contamination kind of worked its way in. So you notice how you have kind of like the background that's nice and light? Do you see that? But if you had lots of little colonies, and when I say that, I don't mean it this way, because that's all Micrococcus. But if you had a bunch of larger colonies, those are probably contaminants. And if that happens with Staphylococcus, and it might have, you can kind of, I hate to tell you to ignore it, but look at it in a very critical way. But the result is you're going to be able to evaluate how well each of your 4 isolates do against a variety of different bacteria. And to remind you, these bacteria are actually relatives of things that cause disease. So in any event, that's what I wanted to tell you there. And I'll probably share a photograph of the background situation with Staphylococcus. I am really, really excited about what you've done. Thank you so much.